All right, kia ora everyone. So welcome to the last of my set of mini lectures that I'll record. And so this is the equivalent of the last lecture that I'll give for this course. And to get started, I'm just going to turn my video off and go straight to the slides. Okay, so what I'm going to do in this and the next few mini lectures is cover the material that will be lecture 15, and this is advanced concepts in fungal phylogenetic relationships and pathogen outbreaks. And so we're going to talk a bit about these two related ideas. So in order to understand the pathogen outbreaks, we have to understand a little bit about phylogeny. So I'll get started. And these are the learning objectives for this lecture. So this comprises all the mini lectures that cover this, not just this one. And so these are to define the phylogenetic terms clade, monophyly, and paraphyly, discuss the phylogenetic relationships of the major groups of fungi, including the placement of the Glomeromycota and the paraphyly of the Zygomycota and Chytridiomycota, describe issues in anamorph-telomorph taxonomic relationships with respect to clades, monophyly, and paraphyly, and how phylogenetic trees can help with the one fungus, one name paradigm. Predict the differences in population structures of pathogens depending on the nature of the outbreak, and interpret phylogenetic trees of pathogen populations to infer the likely nature and origin of outbreaks. So by the end of all this, you should be able to do all these things. So I'll get started talking about phylogeny in this particular mini lecture. And what I'm showing you here is the fungal evolutionary relationships that were covered in 204. And so this is the basic picture showing the Basidiomycota, Ascomycota, the Glomeromycota that we didn't really cover, and the Zygomycota and Chytridiomycota. And what I'm going to do is, if we move to the next slide, is show you the actual evolutionary relationships of the different groups of fungi. And so here's the next slide. And this, you can see that there's a lot more names on this phylogenetic tree here. Down the bottom, you'll see Ascomycota and Basidiomycota. Maybe I'll turn my laser pointer. And here's the Glomeromycota, but then there's a whole bunch of other names. Here's the Chytridiomycota, but there's a whole bunch of other names that you probably don't recognize, and we can ignore the Microsporidia for the purposes of this course. What I've done, however, over here on the right is indicate for each of these names which group from that previous phylogenetic tree that particular group of organisms fell under. And so you can see that there's a mix of things. So this group here is the Zygomycota and the Chytridiomycota. But you can see that most of these just fall into one or other of those two groups that I talked about before. And what we're going to do is we're going to look at how to interpret these phylogenetic trees to see what the outcome of the organization of the fungi into these different groups means for taxonomy, and why we can't just use these simple names, Chytridiomycota, Zygomycota, Glomeromycota, Ascomycota, and Basidiomycota. In particular, how the Zygomycota and Chytridiomycota are not actual real taxonomic names according to the phylogeny. So in order to show you that that's the case, we need to cover some basic concepts in phylogeny. So I'll go to the next slide. And what I want to do is introduce some basic terms. So the first term is clade. So if we look at this phylogeny here, just some basics about phylogenies. So phylogenies are a way of representing the evolutionary relationships between organisms. And so on the right, or at the tips of the tree, these are the extant lineages. And as we go back in time, we go back to more distant populations that existed in the past. And the idea is that when there's a branching in the tree, that represents what was initially one particular population or species or whatever that's now split into two. And so, for example, if we imagine that A, B, C, D, and E here are all different species, then at some point in time, X here, there was a species that split into two different populations that eventually went on and formed the extant species A and B that we see now, and et cetera, if we go further and further back in time. So you can make phylogenetic trees out of kind of any taxonomic group. So these could be individual organisms in a population, they could be species, they could be, you can go as far as different domains of life. You can make phylogenetic trees of different domains of life. So it doesn't matter, you can do them for whatever groups, taxonomic groups of organisms you happen to choose. And the fundamental group of a phylogeny is this term clade. And so that's a group of taxa that share the same common ancestor. So for example, A and B, as I said, they all came from an initial common ancestral population that's indicated by X here. And so A and B form a nice clade that's defined by the split of this branch into these A and B. Similarly, we've got a clade that encompasses C, D, and E that's defined by this ancestor Y. So this ancestral, let's imagine it's a species, let's imagine these are species. So this was imagine this is an ancestral species that split into two populations that went on and formed species C, and then another one that then split again to form D and E, okay? So C, D, and E form a nice clade. D and E also form a nice clade because they are united by this common ancestor here. And A, B, C, D, and E also form a nice clade because they're united by this common ancestor here. So there's not just one clade in a phylogenetic tree. You can have multiple different clades at different levels. But for example, A, B, and C alone don't form a clade because there's no common ancestor that unites them to the exclusion of the other members of the tree.